Hello everyone, today I am going to talk about reconstruction of phylogenetic history to resolve the subspecies anomaly of Pantherine cats. I am Ranajit Das from Manipal Center of Natural Sciences, Manipal Academy of Higher Education, Manipal, Karnataka. Before going further into the topic, I would like to introduce what I mean by Pantherine cats. Pantherine cat includes all charismatic big cats like tigers, lions, leopards, snow leopards and jaguars. All of them are highly endangered and are found in small fragmented pockets around the world. Now, let us introduce the different subspecies of Panthera. I am not going to read the entire slide, but just to give it a perspective, there are 6 subspecies of tigers, there are around 9 subspecies of lions, and but all of this subspecies categorization is generally based on morphology, and most of them are not genetically validated. Now, what is a subspecies? The subspecies definition came way back in 1963 when Professor Ernst Mayr defined it as a fairly permanent geographically isolated race of a species. And this definition, as you can see, is quite vaguely defined and it is mostly based on morphology, but sometimes it can be very important for the conservation of these species. Genetic divergence, basically genetic divergence variable among the species can be called as subspecies. Now, how to solve this so-called anomaly of big cats? Big data can help. What is big data? Big data is basically the data explosion of coming in this genomic era starting from around 2001-2003, and in 2012, as you can see, there is a huge amount of biological data that is available currently in several different databases, including humans as well as other animals. These are some databases that I am showing here. Contains huge amount of genetic and genomic data that can be not only useful for disease studies and conservation, but also it can be used for very basic science research, including population genetics, genomics, phylogenetics and all that. But there is a problem. The problem is basically data management and reproducibility. But if someone can overcome that, this big data can resolve many issues, such as today I am going to discuss about how it can resolve subspecies anomaly in Pantherine cats. What I did was, first I retrieved around 43 Pantherine mitogenomes. Mitogenome, by mitogenome I mean whole mitochondrial genome, that is including all available subspecies from tigers, lions, leopards, snow leopards, jaguars, and I used Neophiles as the outgroup. And this database came from the GenBank, as you can see here. The complete mitochondrial sequences were retrieved by excluding the control region of 15,000, which finally contained 15,000 base pairs. To find out the divergence time, I use Bayesian Markov chain Monte Carlo method, Bayesian MCMC method as implemented in BEAST. What is Bayesian MCMC? Before going into Bayesian Markov chain Monte Carlo method, I would like to tell you what Bayesian method is. Bayesian method is basically comes from the Bayesian statistics, which is shown here. The Bayesian statistics gives you a posterior probability based on the likelihood of an event multiplied by a prior probability of that event, which is shown by this equation. And in Bayesian Markov chain Monte Carlo, there is a Monte Carlo simulation that redefines the data and recalculates the possible divergence time and samples as shown here. For this purpose, as I mentioned before, it needs a prior probability. I used log normally distributed prior probability since it samples more from the distant past than the recent past. The rate of evolution then I recalculated using log normal distributed prior using mu equal to 0 and standard deviation sigma equal to 0.56. After that, after running a Markov chain Monte Carlo, I got this phylogeny. in this phylogeny, in the top you can see the lion, in the bottom there's tigers, and the leopards, snow leopards, and the jaguars are in the middle. It includes all the currently available subspecies, so-called subspecies of all these Pantherine cats. I use 1000 bootstrap replicate to form the tree. Just to give it a perspective, any node with less than 50% bootstrap support were collapsed, and as I mentioned previously, Neophelix cats, which is the closest living relative of this Pantherine group, are used as the outgroup. Individuals with unknown subspecies status are shown with an asterisk. These are the divergence time that I received using Bayesian MCMC method. We found that the divergence between the entire Panthera genera and the Neophelix took place around 8 million years back, and between the tiger and the rest of the clade around 7 million years back, and so on. Just to mention here, the deepest root within tiger came around 2 million years back, and the deepest root within the lions came up around 1 million years ago. Now, how can this help to resolve the subspecies anomaly? As I mentioned before, the subspecies is largely based on morphology, and that is true especially for the lions. What we found is geographically isolated Senegalensis subspecies of lions and Azandica subspecies of lions, along with the Nubica subspecies of lion and Panthera leo with unknown subspecies status, shared identical sequences. Seem like there is no sequence divergence or genetic divergence among these 4 individuals. In contrast, two Senegalensis subspecies individuals, as mentioned here, showed high genetic divergence, which just gives you an idea that the variation that we can see is basically the individual level variation. There is no such race or subspecies going on. So, what we can say here is there is more intraspecific divergence, that is individual level variation, than subspecies level variation, which has to be interpopulation variation as mentioned by Ernst Mayr. The same thing holds for the tigers. The Amoyensis subspecies of tiger and Altaica subspecies of tiger, with this, the GenMAC number is shown here, showed identical nucleotide sequences, while Panthera altaica, 2 individuals, the numbers are shown again here, showed high nucleotide divergence, more than 1%. Again, what we see here is the divergence, the variation that we see is at the individual level, not at the subspecies level. So, probably the subspecies level divergence is not even valid. From here, we can conclude that subspecies status based solely on morphology are largely invalid. We need to revisit this concept of subspecies looking at the genetic data. We need to make a yardstick to say, okay, up to this level we can call it a subspecies, up to this level we can call it a population, and so on. So, since this is not currently done for the Pantherine cats, this variation of the subspecies validation is largely not true. The multiple individuals of the same subspecies is mandatory to resolve the complex phylogeny, such as in case of Pantherine cats, which is also currently not available. With that, I am going to finish my presentation with this quotation from Aristotle: "The whole is more than the sum of the individual parts." Make perfect sense for biology, especially for the big data in biology. Thank you.